fermentation. So the question as to whether fermentation will happen during cellular respiration for us is going to come down to a matter of whether or not O2, oxygen gas, is present. If it is, cellular respiration can continue in the mitochondria for eukaryotes. They're going to be able to use that oxygen to accept the electrons from the electron carrier and the electron transport chain, and will make lots of ATPs. However, if oxygen is not available, then fermentation is an option that can allow the cell to continue to produce energy, though at a drastically lower rates. Plus, again, we are assuming that we are looking at eukaryotic systems here that do have access to mitochondria, and then you do have the option. Of course, if you don't have any mitochondria, then you have no option to actually carry out the rest in the mitochondria, obviously. So, glucose to pyruvate is going to produce some ATP, so that's a great way to make a little bit of energy. And in doing so, these electron carriers are generated, which, if you have mitochondria, or at least electron transport chain, something similar to it, you can then create some more ATP from it. However, that's not the only option. Fermentation is another option. Lots of different organisms carry out in different ways. So we can see things like the bubbles you see in Swiss cheese are due to the production of CO2, and the taste that you get with that Swiss cheese is the acid product produced as well. So certain bacteria are able to do that. Lactic acid, very familiar for things like yogurt, are also produced. You get that lactic taste, that tangy taste of yogurt is due to the production of lactic acid, also carried out by many different organisms. And then, of course, we have alcohol fermentation, carbon dioxide produced, and ethanol as a byproduct. Plus, there are some that can do some other productions, like the production of acetone or isopropanol, and acetic acid is another option as well. So lots of different types of fermentation that can be used. Can use pyruvate to run through this any one of these fermentation processes, and importantly, this electron carrier gets regenerated into NADH, because if you just kept running through glycolysis, you're going to run out of NAD+. And if you don't have any available NAD pluses, then you shut down glycolysis. So fermentation is one of the ways to turn NADH back into NAD+, not in a hugely energetic, beneficial way, without an electron transport chain anyways, but at least it allows glycolysis to continue, because you'd continue to have available NAD pluses, which then can be used to take electrons so you can continue your glycolysis process. So again, we're getting some ATP out of glycolysis, but if oxygen is present, we can make a whole lot more ATP through aerobic respiration using the electron transport chain, and again, pyruvate oxidation, Krebs cycle. Then the electron transport chain, you get lots and lots of ATP. But if that's not available, then we can also run, or cells in general can also run various fermentation processes without that oxygen being present. So again, the term is anaerobic, meaning there is no oxygen available, or at least not enough oxygen available. And again, ATP is not formed during fermentation, but the reactants necessary to carry out glycolysis are reformed. Therefore, glycolysis can continue. Therefore, some energy production is still available. We're going to look at lactic acid fermentation and alcoholic fermentation. So first off, bacteria, plants, you can carry out lactic acid fermentation, and anytime you have overused your muscles, you may feel that burning sensation during use, where you have not enough oxygen present. You're over consuming the oxygen available, and your muscle cells are able to actually switch to lactic acid fermentation as a means of still getting some energy. So this is, again, glycolysis happens. This is happening after glycolysis. We are making two pyruvates, sometimes, and as earlier described as pyruvic acid, and they get converted, instead of normally, into carbon dioxide completely during pyruvate oxidation and cellular respiration. Instead, they run through this alternative pathway where they are produced into lactic acid. We feel this again during exercise, not the soreness after exercise, that's muscle tearing and whatnot, but during exercise you can actually feel this burning feeling, and that can be due to a lack of oxygen and a production of lactic acid. Here's the glycolysis, running glycolysis through, and we produce pyruvate. So that produces some ATP. As long as you have an electron transport chain available, you can use the NADH produced in that as well, and you get some more ATP. But if that electron transport chain is not available or is not able to be run because of a lack of oxygen, then what would happen is you'd eventually not have this regeneration of NADH, which means you couldn't have glycolysis happening, which means you get no ATP. That would be a bad thing. You're not getting any from the electron transport chain, and even glycolysis itself would shut down. So what happens is NADH skips the electron transport chain and instead runs through fermentation in order to regenerate NAD+, so that glycolysis can continue and so that ATP can continue to be produced. Not as much as if you had an electron transport chain, but at least you can have some ATP generated by running this fermentation glycolysis cycle. For us, we run the lactic acid cycle. So this lactic acid, lactate, is itself sort of there temporarily, and again, think of it as a means of regenerating the NAD+ so that glycolysis can continue, so that we can still make some ATP. The NAD+ here is able to be reused, and so this cycle is able to continue. If the electron transport chain is not working fast enough to regenerate its own NAD+, then we can then have this glycolysis continue and still get this ATP molecule. However, we start building up lactic acid in this process. The NAD+ gets produced, the glucose gets consumed, but we start building up more and more and more of this lactic acid, which you feel as that burning sensation in your muscles during overuse. And so what happens is eventually, hopefully, we'll have more oxygen available to us, but essentially we're sort of putting this extra lactic acid into our system, which has to be dealt with later. It can be dealt with by shipping it to the liver, and the lactate is then converted back into glucose once we have more oxygen available to us, because as you can see, this process itself uses up NAD+, plus uses up a whole bunch of energy in order to do so. So it's obviously not a sustainable method. There's no point in making 2 ATPs and then using up 6 to get them back. This is only a temporary measure that is useful when there is not enough oxygen available. The bill comes due later, and that's when we're going to have to get rid of all this lactic acid, and that is what's happened in our liver through this other process here. So this is called the Cori cycle, and this generation of lactic acid as a temporary measure that will then have to be converted back to glucose later on at a fairly high cost in terms of energy. But at least in the meanwhile, you still get some energy production. If you're demanding a lot of your muscle cells and they're being overused, you don't have enough oxygen to keep up with it, at least you can still get some energy at that point in time. And there's the Cories. Alcoholic fermentation, you'll notice happens in things like bacteria and fungi, which have in common that they are not large multicellular organisms. And so while they produce this relatively toxic alcohol, they're able to get rid of it very easily because they're in constant exposure to their environment. Unlike if your muscle cells were to be producing alcohol, they'd be bathing all the other cells you have in your body with alcohol, which would be a bad idea. And so we would carry out lactic acid fermentation instead. But with alcoholic fermentation, the same idea is happening. There's going to be particular conditions where the glucose is going to be turned into ethanol, also known as ethyl alcohol, but ethanol, and carbon dioxide. So the pyruvic acid, pyruvate, is actually broken down into a 2-carbon molecule plus a carbon dioxide, and the 2-carbon molecule is ethanol. So this happens in bread. You have yeast in there breaking down the sugars, and they're eventually able to turn those glucose into ethanol and carbon dioxide. Now the ethanol will evaporate as you cook it, but the carbon dioxide gets somewhat trapped in there, and that's why you get your rising of the bread. You get bubbles in your bread. Same thing happens with alcoholic beverages, or can happen with cheese as well, where you get this production of carbon dioxide bubbles, which sort of build up and produce these holes in the cheese, which apparently mice love. So again, the point being that if you want to carry out glycolysis, you need NAD+, and it gets used up during glycolysis. So if you want to regenerate it so you can continue glycolysis and continue making ATP, you've got to be able to convert that NADH into NAD+. If you don't have an electron transport chain to do that, fermentation is the other method available to you. And in this case, we are converting it into a 2-carbon molecule plus our carbon dioxide, and that is the alcoholic fermentation part where NAD+ is regenerated, so glycolysis continue and ATP can continue to be generated.